Students, now we are going to see about plant cell and animal cell. Now look at the picture. This picture will be helping us to understand about the plant cell and animal cell. This is animal cell, this is plant cell. Most important structure of plants and animal cell, the differences between plants and animal cell is, all plant cells are having cell wall, but animal cells do not have any cell wall. They have only plasma membrane to surround the living organelles. The center they are having nucleus, then the cytoplasms are present at their own. Even in plant cell is also having the same thing. The nucleus is being at the center, remaining all the cytoplasms are present around. Both nucleus and the cytoplasm combined together are called as protoplasm. So main thing about another important differences between these two cells are animal cell and plant cell is chloroplast. The plastics like chloroplast are present in plant cell, but they are absent in animal. Another important thing is vacuole. So this is vacuole. These vacuole which are present inside the plant cell, but they are absent in animal cell. Sometimes even if it is present, it is smaller in size, but it is very larger in size. So when the cells are being matured, the vacuole is being increased and the nucleus is being at the corner. But on the other hand, nucleus of the animal cell is being at the center. But in the mature cell, nucleus are moving to the side of the near to the cell wall. The entire cell will be occupied by vacuole. So the vacuole is surrounded by important a boundary like structure which is called as tonoplast. So the tonoplast is completely absent in animal cell. Both vacuole as well as tonoplast is absent in animal, but they are present in plant cell. Another important thing is centriole. Centriole is very common in animal cell, but centriole is absent in plant cell. Sometimes during cell division, the centriole may be formed. Once again, they may be disappear. Then, in between a plant cell, there is a connection between one cell to another cell. There is a pore-like structure is present. They are called as plasma desmata. This plasma desmata is very common in plant cell. This is absent in animal cell. Then, lysosomes are very rare in plant cell. If we see in plant cell, lysosomes are very rare, but it is very common in animal cell. In plant cell, the storage material is starch grains, but in animal cell, storage material is glycogen granules. So these are the main differences between plant cell and animal cell. Now we shall see the structure of the plant cell in detail. So this is the plant cell. So superficially once I shall explain about the structure of this cell. So this is a plant cell. The plant cell is completely surrounded by cell wall. Inside the cell wall there is a plasma membrane. The membrane which is present around the cytoplasm is called as plasma membrane or cell membrane. Inside the cell membrane there is a place which is present. These are called as cytoplasm. Inside the center of the cell there is a large round body is there. This is called as nucleus. This dark round body is called nucleolus. Around the nucleolus, chromatin materials are there. These chromatin materials become into chromosome later on during cell division. Then around the nucleus, there is a membrane is there. These are called as nuclear membrane. Near to the nuclear membrane, there is a tube-like structure or a network-like structure which are present around the nucleus inside the cytoplasm, which are called as endoplasmic reticulum. There are two kinds of endoplasmic reticulums which are present inside the cell. They are rough endoplasmic reticulum and smooth endoplasmic reticulum. What is the differences between rough and smooth endoplasmic reticulum? Look at this picture. This is ribosomes. So this dark body, round body, white color body, which is called as ribosomes. These are free ribosomes. Sometimes these ribosomes are attached with the endoplasmic reticulum. If the ribosomes are attached with the endoplasmic reticulum, these are called as, we can call it is rough endoplasmic reticulum. So some of the endoplasmic reticulum without any ribosomes which are called as smooth endoplasmic reticulum. Then we can see about chloroplast. Chloroplast is very important organelle which are present inside the cell because this is doing an important function of photosynthesis. So with the help of sunlight, the plastic which is present inside the cell, that is, pigment which is present inside the chloroplast, which is called as chlorophyll pigments, which is trapping the sunlight, which can able to convert this light energy into chemical energy. So this is a major important process which is essential for every living organism, because the chloroplast which is doing a function is called photosynthesis, which is not only producing food for plant but for food for every living organism around the world. Apart from this, during the photosynthesis process, they are releasing oxygen. So releasing of oxygen which will be essential for breathing of every living organism. So another body is called Golgi apparatus or Golgi complex. So these are present inside the cell. Then various, as I said just now, there is a vacuole is present. Inside the vacuole, lot of chemical substances which are present inside. This vacuole is surrounded by a membrane called chloroplast. Another important organ is called as mitochondria. So mitochondria is also called as powerhouse of the cell because they are doing respiration in function. Because of this respiration, this energy will be produced inside the cell. So that is why mitochondria supply the energy for living organisms. Hence, these are called as powerhouse of the cell. Another organelle is called as peroxisome. This peroxisome is doing photorespiration. So in such a way, various organelles which are present inside the cell, which will be helping for living organism, planet. One by one we can see. First of all, we shall see about cell wall. The cell wall is the outermost protective covering. So this is cell wall. So this is outermost protective cover of the cell. It is present in bacteria, fungi and plants as well. Whereas it is absent in animal cell. It was first observed by Robert Koch. It is an activity growing portion. It is made up of different complex material in various organisms. In bacteria, the cell wall is composed of peptidoglycan. So cell wall is in bacteria, the cell wall is made up of by a specialized chemical is called as peptidoglycan, which is made up of N-acetyl glucosamine and N-acetyl muramic acid with various amino acids. So that is called as peptidoglycan. In fungi, the cell wall is made up of by chitin. Then fungal, as well as in fungal cell, in almost all the fungus, this cell wall which is made up of by cellulose. In algae, which is made up of cellulose, galactose and manganese. But in higher plant, it is made up of by cellulose, hemicellulose, pectin, lignin, cutin, suberin, silica. So these are all the chemicals which are present. Then inside the cell, which is made up of by cell wall, the cell wall is having three different walls. They are called as primary wall, secondary wall and middle lamellae. So we can see about the primary wall, secondary wall, middle lamellae inside the cell with the help of electron microscope. So what is primary wall? If you see middle lamellae, look at the picture, I shall show you. So this is cell wall, this is middle lamellae. Inside the middle lamellae, there is a wall is formed. This wall is called primary wall. Then inside the primary wall, next layer is called secondary wall. The secondary wall is producing as many times as possible. These are further divided into secondary wall 1, secondary wall 2, secondary wall 3. So inside the body is called lumen. It is not originally lumen. Inside the lumen, cytoplasm and nucleus will be occupied. So this is a cell, this is neighboring cell. How these are? In between the two cells which are connected by plasma desmata, there is a chamber or opening which will be called as plasma desmata, as well as simple and border pits. Now we shall discuss in detail about the primary wall. So just below the middle lamellae, it is present. So it is first layer into the middle lamellae. Primarily, the primary wall is consisting of loose network of cellulose microfibril in a gel matrix. It is thin, elastic, extensible. In most of plants, the microfibrils are made up of cellulose oriented differently based on shapes and thickness of the wall. The matrix of the primary wall is composed of hemicellulose, pectin, glycoprotein and water. Hemicellulose binds the microfibrils with matrix and glycoproteins control the orientation of microfibrils, while the pectin serve as a filling material of the matrix cells such as parenchyma and meristems. Then we can see about the secondary wall. After the formation of primary wall, secondary wall will be formed. The secondary wall is laid during maturation. When the cells are beginning to mature, the secondary wall is formed. It plays a key role in determining the shape of the cell. It is thick, whether the shape is circular or rectangular or polygonal. This will be depends upon the formation of secondary wall. This secondary wall is very thick, inelastic, and it is made up of cellulose and lignin. Secondary wall is divided into three sublayers called S1, S2, S3, where the cellulose microfibrils are compactly arranged with different orientation, forming a laminated structure on the cell wall. Now we shall see about the middle lamellae. The middle lamellae might be formed during cell division. It is outermost layer of the cell which is made up of calcium and magnesium pectin deposited at the time of cytokinesis during cell division. So cytokinesis means during cytoplasm is divided during cell division. It is a thin amorphous layer which cements up to adjacent cell. So the two adjacent cells are cemented by this amorphous material of middle lamellae. So in between the plant cell, these are connected by plasmodesmata and pits. Plasmodesmata act as a channel between the protoplasm. That means inside the protoplasm. That means this will be penetrated through middle lamellae, primary wall and secondary wall as well. So there is a channel-like structure which is formed between the two adjacent cell is called as plasmodesmata. So which is forming adjacent cell through which many substances which is passed through. Moreover, at few regions the secondary wall layer is laid unevenly, whereas the primary wall and middle lamellae are laid continuously. Such regions are called as pits. The pits or adjacent cells are opposite to each other. Each pit has a small opening, a pit chamber and pit membrane. The pit membrane has many minute pores, small hole-like structure, and thus they are permeable, which is allowing only a certain substances to pass through them. The pits are two types, namely simple pits and border pits. If secondary wall is surrounded, it is border pits. There is no secondary wall is surrounded, then this is called as simple pits. Then the middle lamella, primary wall, secondary wall, plasmodesmata and the simple and border pits, these are all combined together will be forming cell wall. Then what are the functions of this cell wall? The cell wall offers definite shape and rigidity to this cell. The cell wall serves as a barrier for several molecules to enter the cell. The cell wall provides protection to the internal protoplasm against mechanical injury. The cell wall prevents the bursting of the cell by maintaining the osmotic pressure. It plays a major role by acting as the mechanism of defense for the cell. Next we are going to see just below this cell wall there is a membrane is present, that is called as plasma membrane or cell membrane. Now we shall discuss about cell membrane. The cell membrane is also called as cell surface or plasma membrane. It is a thin structure which holds a cytoplasmic content called cytosol. It is extremely thin, lesser than 10 nanometer. We can see there is a fluid mosaic model of plasma membrane that can be seen through the picture. So this is fluid mosaic model of cell membrane. This was given by Singer and Nicholson. The fluid mosaic model was given by Singer and Nicholson. It is made up of by lipids and proteins. We can see this structure. If you see, it is channel protein. This is called as alpha helix protein. Here the dot on this channel protein, there is a dot like structure, it is called globular protein. Here this is glycoprotein. This is the dots is called carbohydrate on this protein. Then glycolipid on this protein. Then it is called cholesterol. It is integral protein. It is peripheral protein. In such a way, it is made up of by lipids and proteins. The yellow color part, which are called as lipid, these proteins are embedded. They are combined together to form lipoprotein, that is mosaic model. So Singer and Nicholson proposed this fluid mosaic model. It is made up of by lipids and proteins together with a little amount of carbohydrate. Lipid membrane is made up of phospholipid, that means which is made up of by phosphorus and lipids. Lipids means fat. Both phosphorus and lipids are combined together to form phospholipids. The phospholipids molecule has hydrophobic tail and hydrophilic head. So this is called as phospholipid material. Now look at the picture. It is head, it is tail. It is head, it is tail. So both the sides are having head and tail. This tail is called hydrophobic tail, and the head is called hydrophilic tail. What do you mean? The hydrophilic tail repels water. The hydrophilic tails will not be connect or join with water. They repel against water. But on the other hand, the head is hydrophilic in nature. So hydrophilic head attract water. The protein of the membrane are globular protein, which are found intermingled between the lipids. It is, the lipid is bilayer. Here and there, the protein is intermingled. So the globular protein which are found intermingled between the lipids bilayer, most of which are projecting beyond the lipid layer. That means the lipid layer are being here, some of the proteins are projecting outside. These proteins are called as integral proteins. Few are superficially attached on either surface of the lipid bilayer, which are called as peripheral proteins. So if it is being outside, they are called as peripheral proteins. If it is being internal, they are called as integral protein. The proteins are involved in transport of molecules across the membrane. Any materials are transmitted through the membrane, this lipoprotein will be helping them to transmit. So they do not allow all the materials to pass with them. Only a permeable material can be passed with them. The carbohydrate molecule of the cell membrane are short chain polysaccharide. These are either bound with glycoproteins or glycolipids and form a glycocalyx. The movement of membrane lipids from one side of the membrane to the other side by vertical movement is called flip-flopping movement or flip-flop movement. This movement takes place more slowly than lateral diffusion of lipid molecule. The phospholipid can have flip-flop movement because the phospholipids have smaller polar region, whereas the protein cannot flip-flop because the polar region is extensive. So this can be seen how the transport of molecule through cell membrane with the help of a diagram. So it is not shown. So that can be seen later on.